Hi guys, welcome back to the 8th episode of This Is Not News. I'm your host Jai, and today on the episode we have Wytamma Wirth. Now, Wytamma is a virologist up in Townsville. He's actually doing his PhD at the moment, specifically working with turtles. We do talk about that a little bit in this episode, but we sat down today. We actually also went live on Facebook just to get some more information about coronavirus. There were some questions that I wanted to know, and I also opened it up to a lot of people in the This Is Not News group and the wider community to send me questions, things that we want to know about coronavirus and how it works and interacts. So this was a really great chat with Wytamma, and I think we should just get straight into this one. So I hope you guys enjoy it. All right, you all right, ready to go?
Yep. Yeah, good. Got my glasses on. Got my Barra shirt.
Sweet.
Smart.
Dope. So, how you been, man? What's been going on?
Yeah, like I said, I've just been about to finish my thesis, my PhD. So I've been doing basically a lot of uni, which is like I guess since we met back in uni hall 10 years ago, something like that. So basically just haven't left since then and just stuck it out at uni.
Yeah, true. So what you've still been, so when you were back at uni, when we knew each other, when you're doing, I suppose, your bachelor or your undergrad, what were you studying then before you?
Biochemistry.
Yep. And so then have you gone back and done other stuff then afterwards?
Yeah. So I did an honors degree in microbiology and immunology after that. Yep. And then I've started my PhD now in epidemiology and pathology in wildlife disease, actually.
Oh, cool. So is that, I suppose that's semi-related to coronavirus then, isn't it? Because
it's all sort of part of it, I guess. Like the modern sort of approach of virology is this like One Health idea. And so that all the wild animals, the environment, and human health is all sort of linked together.
Yep.
So each aspect of that is really important in sort of this like disease outbreak, and like sort of obvious with coronavirus. If the bats weren't there, like there are obvious wildlife component to this.
Yeah, cool. So let's start off by just tell everyone who you are, what you do. I suppose we sort of went over it a bit, but tell everyone what you do and where you are at the moment.
Right. Yeah. So I'm Wytamma Wirth. I'm a PhD student in Townsville in JCU in northern Queensland. And I work on wildlife disease, mainly focused on the pathology and epidemiology of coronavirus infection. I work closely with turtles, actually, and we do a bunch of other work on different animals and some bird flu work, as well as different sort of wildlife disease aspects, disease investigations in wildlife.
Yeah, cool. So what's been going on up there then? Like obviously you're still in the academic world. Have people sort of what's happening in the academic world in terms of virology and that at the moment? I imagine it's
so we were
hectic.
We were pretty I guess lucky that we have a bunch of academics who are very interested in this thing early on. So they sort of made the transition to online learning fairly rapidly within our college. We're in a public health college. And so they were sort of switched on to this thing being a problem and made sure they went through a training of like taking all the classes to online learning. So that was pushed through fairly quickly, and all the classes transferred to online learning. And they've basically, the campus isn't shut down. You can still go in there, but they're sort of saying if you don't need to come in, don't bother. Yeah. So everything is transferred to online. Which has been good. It's actually had a reasonable social component to it because that it just basically has created these Zoom meetings to keep everyone up to date. And so people who wouldn't normally, a PhD or a high degree by research sort of program is fairly isolating. You're working by yourself a lot, but now everyone is sort of joining in bi-weekly or weekly to these meetings with everyone. So it's actually been fairly social, which has been good.
That's interesting that like it's sort of gone the opposite way for you guys. It's gone from being, to actually being more social just because of everything going on.
We actually started a research student Facebook chat, which we didn't have before. No one just talked to each other. There's an actual chat where people are communicating. So
do you guys share like articles and stuff to do? Because like are people in this, in the academic world, even if it's not their specialty, are you guys all sort of still talking about it amongst yourselves and interested in it?
Yeah, it's definitely the talk of the day. And everyone is sort of getting onto it whatever way they can, I guess. So like my housemate is an ecologist and he studies bat movement populations, sort of tracking bats, camps and around. And so sort of he just decided like, well, I mean, this is a huge component for bats, and he can sort of apply his ecological understanding to this. And so everyone is sort of moving in from all different fields to sort of help, um, in this outbreak.
Yeah, cool. That's interesting. I might have to chat to him at one stage. That sounds good. So apart from like that, what's Townsville like at the moment? Because I haven't been up there in a couple of years now. But what's going on in terms of? I mean, you're probably not going out much, but have you heard much about what like how it is in Townsville at the moment?
Yeah, well, it's still reasonably hot up here. It's slowly starting to cool down a little bit, but as you know, it's in the tropics, so yeah, it's been fairly hot. They've basically they started to close down. They initially started to close along with the rest of the state, I guess, as like pubs and large gatherings and stuff closed. They've recently started closing things like the goat track. So there's a big hill
Yep.
in the middle of town, and so up Castle Hill has been closed down. Basically, people were too close to each other on the track. But
Yep.
we haven't had a case in 2 weeks up here, so it's. I know some of the local politicians are sort of calling for a isolation of North Queensland to put uh, a border in at Bundaberg somewhere or something like that. And
Yeah, the
saying,
old North Queensland argument. They always want to make another state up
exactly, yeah.
there. Yeah, that's crazy. Yeah, so there hasn't been many cases up there then, hey, have there?
No, there's a total of 24 cases, I think.
Yeah, cool. Well, that's good then. Good, it hasn't affected you guys too much. So, I've been sending you. You've almost been sort of my coronavirus fact checker for the last couple of weeks or so, just because it's like, oh man, what about this? I just heard this, or send an article to you, and you've usually already read it and can tell me sort of what's going on. So maybe we sort of start with things that we definitely do know
Hmm.
about it so far. So I remember I was talking to you about like how long it lasts on surfaces.
Yeah.
Um, and I remember you sent us that research article now. So what are the different surfaces and how long it sort of stays there from what we know, and like what's the difference between those surfaces and why, can you give us a little bit of maybe why those surfaces, it acts differently?
Right. Yeah. So, yeah, there are like there's a range of different surfaces that were sort of tested in that study, and they tried like metal and plastic, wood and cardboard. And I guess there's different sort of inactivation properties of these surfaces. Things like copper have like a sort of known inactivation property, and it's the research has basically come from antimicrobial studies in bacteria. Um, uh, but basically, I think the main sort of takeaway with these coronaviruses, because they are enveloped viruses, is how well the moisture can sort of hold on these surfaces. So something that can dry out a lot quicker or hold moisture, like cardboard, can kind of be a bit soggier, so it might not last a bit longer on there. Things like plastic have little grooves where it can sort of hide into that area. But yeah, the results have sort of said that it could last up to weeks on some of these surfaces. But a lot of these studies haven't really been using infectious virus as a measure of whether this virus is still viable. So even though you might detect the virus on there, it's not actually a measure of whether the virus is active, is there in a state where it could infect you afterwards.
Yeah.
So I've heard the analogy of like if you detect dog pee on a fire hydrant, it doesn't mean there's a dog there. Like there's part of the dog or something like that. You can
Yeah.
still another dog can go and smell that fire hydrant and detect that there was a dog there,
Yeah.
but it doesn't necessarily mean the dog is still there. So the same with these sort of tests. It's hard to say. Generally, these viruses are not very stable at all. Envelope viruses will not last that long on these surfaces. Yeah.
Yeah, because I think there was an article I sent you, and it said something like they found remnants on a cruise ship like 17 days after people had been off the ship. And that's what you sort of said to me. Well, it's like you can find some of the RNA or whatever it is, the
Mm.
genetic material of it, but is it infectious? And so I suppose it's easy to blow that up in the media then and go, oh, we found it.
Exactly. Yeah. Yeah. It's definitely a common pitfall, and it happens with because you can't have infectious virus without the RNA, but you can have RNA without the infectious virus. So it is a trade-off, and these are very sensitive assays. You can detect really small quantities of these viruses or the RNA that's left there.
Yep.
But you don't. Yeah, exactly. It doesn't tell you anything about the infectious state. So you have to interpret. That's one thing I guess that's coming out of this is interpreting everything in context, so you actually know what you're looking at.
Yeah. And then I remember there was also an article saying, and it's probably a similar thing about how it can stay in the air for hours and stuff like that.
Right. Yeah.
Did you read
Yeah.
that article as well?
Yeah. So that was so basically they used a specialized type of apparatus in the lab that basically keeps the virus floating in the air. So they would measure it in the air, but it's not like the virus would actually stay in there in a normal environment. Normally it would just fall out of the sky because they're large enough that they would just fall out of the air. But in this special apparatus, they're basically testing whether the air would inactivate it without it touching any other surface. So
Okay.
it lasted 3 hours, I think, in the air like that. But it doesn't mean that it will actually stay in the air. It's just
Yeah.
if maybe, I don't know, it was in a space station or something where it wasn't.
In the space station.
Yeah, might be a
Let,
problem.
let's hope it doesn't get to the space station.
Yeah, exactly. The last was
That could be a problem. Yeah, cool. So, has there been any other interesting research maybe you've seen recently, or that sort of caught your eye where you went, "Oh, that's interesting," or something that you thought was quite interesting?
Yeah, so I think some of the interesting stuff is the vaccines that are coming through. So there's been a big push um, for to find a vaccine. Basically, that's sort of one of the only sort of hopes, I guess, that we have, apart from sort of drugs for treatment of cases. But as a prophylactic, yeah, vaccine is probably the way to go. And I think currently there's over 100 vaccines that are in various stages of clinical development, and then yeah, 5 or 6 of those are actually in phase 1 clinical trials. So they're actually putting them into people. And I think one of the interesting vaccines from that are these RNA vaccines. So basically, they are a vaccine that you can give someone, and then the body will produce the vaccine. So you don't have to grow a virus up in culture like the flu vaccine or something like that and give them a weakened form of the virus that will still replicate. This is just the genetic material, and they give that in a little lipid body, and that cell will take that up and then make the protein inside the cell. So you sort of produce the vaccine yourself in your body, and that's sort of the idea, is that that'll sort of provide better cell-based immunity compared to some of these other just protein-based vaccines, and it's much easier to develop than these inactivated virus or attenuated virus vaccines. And it was really cool because they could take basically from when the sequence was published in January, early in this year, they basically just downloaded that sequence from the internet and then put it into a machine that prints out the RNA, makes the sequence, and then that's the vaccine basically done. So they could just, from one side of the world to the other, from China to, I think these are an American company, the Moderna RNA. They developed this vaccine basically in 2 months or a month or something like that. They had a vaccine ready to go, just printed it out.
That's crazy. So they printed the RNA to make the vaccine. Like,
Yeah, and so basically, because you can just do it the same way you would make the PCR test, you can just produce some RNA that way, and then yeah, that one, if it's the right sequence, they can put it inside a little bit of a lipid bilayer that the cell will take up and eat, and once it's inside the cell will make the vaccine basically itself. Yeah.
that's so cool. That's so awesome. I hadn't heard of that yet. How so they've been working on that for a while, you said then? Like,
Yeah.
is that,
So
yeah.
initially, I think they started working on it as soon as the sequence was published, basically. So they have spent years setting up basically the tooling, so the system to produce these vaccines. But now that they have that in place, when there is a new sort of disease outbreak or something like that, they can just get the sequence from the internet and then put it into their pipelines, and out comes a vaccine. Um, there's some sort of they're not too sure how well it will scale up. So basically, it's hard to produce these at high volumes. But some of these vaccines are actually able to they're like self-boosting. So when you give someone a little bit of it, each little bit of RNA will produce multiple copies of the vaccine. So you only need to give, people are talking that grams would be enough to inoculate millions of people. So it's really sort of concentrated stuff. But whether or not that pans out, I guess we'll see.
Yeah, it's almost a race to see who gets there first, isn't it? Like you said, there's
Yeah,
hundreds of ones.
yeah. A lot of these big pharma companies are actually investing now into like they're making the vaccine now. So they have these, maybe not necessarily that RNA-based vaccine, but some of these other protein vaccines or the more traditional vaccines. They're already producing the vaccines before the trials have even gone through, so that if they do work out, they'll be ready to go straight away as like an investment. Yeah.
That's awesome.
Mm.
That's so good that Yeah, that there's so many different, places or companies trying to once I suppose it's going to be a big profit thing for them as well because everyone's going to want to buy it if they're the first.
Yeah.
So, but that's great to hear that there's so many people working on it and sort of planning ahead for like, hey, if this does work, we're going to be ready to roll it out.
Mm.
That's really promising. What do you reckon for timelines still? Because I know we always talk about this, and it's what's your thoughts?
Yeah.
Obviously, it's hard to tell, but best
Yeah.
guess, what would you say?
I was watching a lecture today from a vaccinologist who was sort of looking at different vaccines, and they were saying that once they have sort of good phase two data, so they've put it into people in like a healthy select group of people, and they can say like, okay, this is a safe vaccine. No, this small group of people hasn't developed any sort of clinical signs from just the vaccine itself, and then they go to a targeted population that they think could be at chance of getting disease, that bigger group of people that might be at risk of developing COVID-19. And so they give that to them. And if they see good like efficacy of the vaccine, that these people don't develop the disease, then if they start ramping up from that point, it's still 12 months away.
Yeah,
So yeah. So from that, once they have this good, then yeah, the green light is to go. They're saying yeah, still a year from that
yeah,
point. So it might be that's for I guess enough vaccine for everyone. So people like healthcare workers and stuff like that, they might actually be selectively chosen to go into these early trials, basically to say like, okay, you are someone who's more at risk. And so just through compassionate use, they'll say, okay, we're going to select you for one of these sort of early doses. So that it might actually not take that long. It'll be I guess, for a regular Joe to get a vaccine could still be a year. But for people who are on the front line, it could come a lot quicker, I guess.
yeah. Cool. I suppose they would just select them for the trial, for the research. Yeah.
Yeah.
Yeah, cool. That's awesome. So then, like, I've had a heap of questions from people, and people talk about like temperature, for example. So I'm just wondering what temperature, like environmental temperature. So obviously we're coming sort of into winter in Australia.
Yeah.
How will that affect the way this, I suppose, behaves? Whether that's how it transmits or how long it lasts in the environment. How does that affect things?
Yeah. I guess like the traditional seasonality with flu is like there's different explanations for it, I guess. But one of them is that during winter, people sort of are indoors, crowded together more, and they're more likely to transmit the virus. I guess it doesn't really make sense for us in the southern hemisphere, who in winter it's a nice time to go outdoors and sort of actually get out from the aircon. But yeah, another thing is definitely the effect of temperature on the viability of the virus. So these viruses, they're enveloped, they're not as stable. They will break down over time in the environment, and that temperature should change that. But it might not, like basically the temperature that we're seeing, because we have had these outbreaks during summer already and through winter in the US, it sort of seems like that the at least the effect of temperature is not that much on the virus. That maybe through like if in a social distancing situation where people aren't coming in contact with each other as much, the virus might be able to stay a bit longer and be more likely to be picked up. But in the sort of non-lockdown situation where everyone's sort of moving around, the effective temperature isn't that much on it. So you wouldn't really see that that effective temperature on virus transmission. So I think that the effective temperature is low enough that we're really not seeing that much of a difference from it.
So they last longer. Like, so then what's their ideal sort of temperature range that they like then for, like, is there a
Yeah,
range there?
I'm not entirely sure for coronaviruses, but it will depend on the virus. There's sort of two different states, I guess, for a virus. There's the infected cell and the virion, or the virus particle. And so each sort of state will have different characteristics. So once it's in the cell, it will have an optimal temperature for replication. And when it's outside the cell, it might have like when it's just a particle, then it might have some optimal temperature for transmission. So definitely, like I work with a lot of ectothermic vertebrates, so cold-blooded animals, and these animals will have their body temperature will change a lot, and so they are able to sort of move around an environment to change where the optimal temperature of the virus would to replicate. So we have like 37 degrees body temperature. So a lot of our viruses are sort of adapted to that replication temperature. Uh, but outside the host, like a high temperature can definitely inactivate these viruses. Um, Low temperatures are generally good at storing them. Like we keep virus in the fridge or freezer or something like that if we're in the lab to
Yep.
store them. But yeah, I'm not too sure how much it will actually have an impact on the sort of epidemiology and transmission of viruses.
Yeah, and I suppose especially because you, like you said, it's been in the northern hemisphere already, where it's been in cold conditions, and I suppose it's I don't know. Has there been much difference between it up there, like how it's transmitted up to the northern hemisphere compared to southern hemisphere? Have they looked into something like that?
I mean, I guess there is the data there from like these different countries, but it's also hard because a lot of them um, basically, from what I've seen, these sort of tropical countries like Vietnam and South Korea and sort of places like this and China, they had extensive tracing, testing, and lockdown, mask use, all these sort of other interventions that we used at the same time. It's very difficult to tease out whether that, like the climate or something, was causing this sort of disease outbreak,
Yeah,
or it was these other sort of aspects that they were putting in place that sort of reduced it. So there has been, like in some of these places, there have been less cases. Like in Australia, we've had less cases, and it's been warmer than in other countries. But it's hard to say because we also locked down the borders. We implemented this very strict sort of quarantine protocols. So yeah, it's difficult to say the effect of temperature.
yeah. I suppose it's hard because every country and everywhere is doing a different, implementing different measures to try
Yeah,
and sort of, stop the spread. So it's hard to say, well, is it the measures that they're doing that's stopping it, or is it the environment, or is it the population density? Like, there's all these factors that
yeah.
make it really difficult, doesn't it?
Yeah, it's thousands and thousands of poorly designed experiments. So like normally you would change one factor at a time, but this
Yeah.
is, yeah, lots of factors sort of changing. So it is difficult. I'm sure it will be analyzed in great detail after this is done, and people are trying it at the moment. But yeah, it's hard to work out what exactly is the cause.
It's like you got to get all the data. Really, we don't really have all the data yet, do we? So it's
Exactly. Yeah.
difficult to figure that out. So, like one question I've had from lots of people and some people, and I think it's important. So maybe just talk about what the difference is between, like, say, this virus to the flu. Like, what makes it different from the flu, and why do we have to sort of the measures that we put in place, why are they different to, what we would do normally with the flu? Because you see people say, oh, the flu kills so many people and stuff like
Right.
that. But how's it different?
Yeah. Okay. So, well, I guess the healthcare system is set up to handle the flu basically because these are very predictable, I guess, outbreaks. We get a flu season, and we get this cyclic outbreak every winter. There is an outbreak of influenza virus in each hemisphere, and the current system is set up for that. So we have, you know, basically, I guess the government doesn't really like to waste money, so they will, yeah, I guess they'll have some sort of capacity for beds, and that capacity includes the seasonal variation in influenza. And so if an outbreak like this happens, where we suddenly double our capacity, hospital requirements, the influenza isn't going anywhere. It's still there. And this is sort of an extra thing on top. I guess maybe if we swap them out, it might not be as bad, but that would be, I guess, if all other things were equal, and you could swap them out, then it wouldn't be bad. But it's because we have two things at the same time.
Yeah,
I think it's also because we don't have a vaccine, so we have a completely naive population. So the virus is able to spread unchecked, and there is no sort of immunity at all within the community. Whereas with influenza, we have good flu system. We do a lot of surveillance. We develop vaccines every year to help mitigate this disease. From what I've seen, this virus is actually more pathogenic than influenza. So for whatever reason, it's the pathology that's caused by this virus is more severe than influenza virus, and so uh, yeah, I guess that is a thing to sort of point to when people say, okay, it's just like influenza. It's like, well, it's not like just like influenza. People are developing different types of pneumonia, and there are all these sort of secondary complications like blood clots and kidney failure and heart disease. So it does seem more severe in terms of just the disease. Yeah.
yeah. So what sort of causes those side effects then? Like when a virus like this enters the body and starts replicating and stuff like that, what actually causes these? I suppose they're side effects, or we would call them symptoms.
Yeah.
But really, I suppose they are side effects of the virus replicating in the body. What maybe causes that in this virus? Do you know much about that?
Yeah. So uh, there is some initial disease that is sort of from the replication of the virus itself, but they think that the majority of disease in this virus is immunopathology. So it's induced from the host immune response. So as we try and fight this virus, we sort of develop an overactive immune response, and that leads to some damage to tissues as the immune system sort of goes a bit out of control. And so a lot of the treatments are actually immune suppressants that they're using to treat this virus. Things that are sort of blocking cytokines and sort of reducing inflammation through this disease. Blood clots are another sort of problem that they've seen come up, as well as heart disease. So the receptors that the virus binds to are also found in cardiac tissue in the heart as well as in the lung. So this sort of it has the ability to go widespread, I guess, within the body. And after the initial replication, people are then seeing this immune response, cytokine storm, and a really strong response to this virus from our immune system, which is then leading to a lot of damage to tissues. Yeah.
Yeah. So then, this might be a silly question, but does that mean it's good to have not a very good immune system for this?
Well, so it's sort of interesting. So I think that it's good to have not a good immune system at the right time. So the immune system is still very important, and they sort of found this with their initial use of steroids for the treatment of this disease. So originally they were giving people steroids when they came in with this disease to sort of tamp down this immune response. But it seems that the initial immune response is actually important in sort of controlling the virus, but it's when that immune response gets out of control that you need to bring it down again. So it's the immune response is still, it evolved for a reason. It's still very important in controlling disease, but yeah, it can still get a little bit out of control. Yeah.
Yeah, so it's just about having it at the right time. That was just
Yeah,
a bit of
the
a silly
balance.
question, really. So then, like, also like with pre-existing conditions. So is it, we talk about pre-existing illness. Is it really any illness, or is it stuff that's to do with where we see damage from it? Like, is it more if you've got things with, so like you're talking about the heart, kidneys, lungs, things to do with that, or is it widespread? Can be like anything. Like, how does that affect things?
So I think definitely things that might limit your oxygen consumption. So exacerbate this sort of problem. So sort of chronic pulmonary diseases and heart conditions are majorly associated by a comorbidity associated with like COVID-19. So I think, yeah, definitely there are specific diseases. So during the pathology, having one of these sort of organs already compromised is going to just lead to worse outcomes, definitely.
Yeah.
I haven't seen too much. I guess like immunocompromised people are not a susceptible group just because they're more likely to get this virus at the start. And if it is able to replicate unchecked, that's a bad sort of thing. Like you will need the immune system to bring it back down and like initially. So it might be good to not have a big immune response later on, but definitely at the start, it's an important part of fighting off this virus is to stop it developing. So immunocompromised people for sure. And basically like humans exist in a state of homeostasis, like all our sort of systems and organs, they're not really, I mean, there is redundancy within our body, but they all play a role. So if you are compromised in some way, it's definitely going to affect your outcome, but just because it's part of this larger system. Yeah. Your
Yeah.
body is investing into some other healing process, then it might not be as effective in fighting the virus.
Yeah. Cool. So what about like reinfection? Is that possible? Is there much evidence, or has there been much research on that? Because I've seen things where they say yes, it is possible. People don't have the antibodies, or there's mutations or things. So what have you seen in terms of like reinfection?
Yeah, so I think reinfection is definitely possible. In China, they've seen a bunch of people become reinfected, but I think that would sort of be expected that people who have SARS or the other coronaviruses or the common cold sort of coronaviruses, they still get reinfected every year, but they just develop milder symptoms. So basically, I think the immune response in this case is not a neutralizing response. So it doesn't completely stop any sort of infection, but it helps the initial immune response stop that infection from developing into something severe. So definitely reinfection is possible, but it basically won't result in this sort of severe acute respiratory syndrome that you see developing in people who are infected for the first time.
Yeah, but you could then be a carrier who maybe spreads it, right?
Yeah, well, I guess that I'm not too sure if they've looked at spreading from the secondary infections, but generally the incubation time at least is shorter. So I heard some people sort of say that these common cold coronaviruses that we have circulating at the moment, everyone sort of gets infected with those, they develop a cold, and they move on with their life. That's not a really big problem. I guess when those viruses sort of initially moved into the human population from animals, these were also animal viruses that moved in at some point. These probably spread around and caused a lot of disease within the human population. But once everyone was infected at least once, it's then only new children who are exposed to that virus. And from what we've seen with children don't really develop a severe disease from this. So we can sort of get into a situation that once it's spread through the adult population, the only new susceptible people will be new children, and they don't develop severe disease. And then once they've had it once, they'll have this immunity and they can get exposed and continue to develop a less severe disease. So it might just eventually reach this point where it finds a sort of niche within our population and continues to circulate, but doesn't cause as many deaths, or if any.
Maybe kind of like chickenpox, like, that's something that, you get in the young populations. Yeah, that's, maybe a good analogy there.
Yeah, it probably chickenpox has like a more of a lifelong immunity, I guess. So you can develop a long-term immunity to this
Okay, yeah.
infection. But like you'd still sort of expect people to develop some sort of symptoms with this one. They may be infectious for a short period, but yeah, as much as just a cold or something like
Yeah.
that.
And so is this something that could take on potentially like a seasonal effect? Something where, we have seasonal coronavirus every year?
Yeah. In the same way that the common cold coronavirus it's like cold season. Yeah, definitely.
Yeah. Yeah, cool. So, what's if we're looking at, say, a year at the earliest for a vaccine, what do you think the next maybe year or 2 years looks like? Just in your opinion, obviously this is we're speculating here.
Right. Yeah.
What do you think's going to happen in this next year or two, while we sort of develop this vaccine before it rolls out?
Yeah, I saw someone on Twitter posting that this is the part of the movie that would be a montage. So that where everyone, I guess there would be a cut scene with Rocky or something, or Eye of the Tiger playing in the background, and some scientists looking in test tubes and down a microscope. And then a year later, cut to the vaccine and everyone's safe. But I guess, yeah, what do we do? What does everyone else do during that time?
Yeah.
There's been talk of, I guess, so there are still exit strategies that people are talking about for this disease. So basically, we can continue this lockdown, I guess. So we stay inside, which is not too fun. It's not good for the economy, I guess, but it stops people getting disease. We've been very successful in Australia in keeping this virus sort of from spreading. We can try suppression. So that would be like allowing specific age groups or non-risk groups or essential workers to sort of come back and start releasing sort of small communities or populations within the community to go out and do these things to sort of help stuff move along. And then people, I guess, like these are sort of strategies that you basically to try and keep things as much as normal until the vaccine comes around. Yeah. So yeah, basically, I think the government's initial plans with these lockdowns were based on modelling from University of Sydney in New South Wales, and so they had said that it would take 90 days or so for this lockdown to completely remove the virus. But once the lockdowns were removed, it would basically come back.
Yeah.
So I think we might see a sort of network develop between countries that are also removing the virus. I've heard talk that New Zealand has offered to recommence travel between New Zealand and Australia.
Okay.
So we might start to build this sort of network of safe countries that it's okay to sort of travel to. So that was sort of the main case, I guess, in Australia. This problem of imported cases was sort of the main problem.
Yeah.
And so if we can identify these countries that are virus free, we can slowly start to reincorporate them. And basically, it would just be until we get a vaccine. So um, there's the chance of herd immunity is not good,
Yep.
and basically we'd need too many people to be infected, and with the current infection fatality rates, it's just not worth it.
Yeah, a lot of people have died because I've seen the research articles or the news articles, I guess, that say 60% of people would need to get it. And then when
Yeah,
you look at the mortality rate, like that's a huge number of people, isn't it?
yeah, yeah, exactly. Yeah, millions of people dying. So it's just not practical in any way. And they can. So the original ideas were like, if we can keep the replication rate or the reproduction rate of this virus down to like 1.2, so that every person infects 1.2 other people. So it's not barely transmitted to two people
Yep.
each person. Um, and so uh, if they were able to do that and keep it at 1.2, the peak would still be in 2021, mid 2021, and the infection would still be going on by 2022, and that's about when we would reach herd immunity. And so it
Yep.
would be this sort of drawn out process to keep people out of hospital, so we don't overwhelm the hospital system. But it would flatten this curve and take it out much longer. At the moment, they're estimating that the reproductive rate is around zero point 0.8, which is why the curve has sort of dropped down.
Yep.
But that's because of these strict measures that they've put in place, and obviously they're not sustainable. So it's going to take, sort of people releasing at these slow times. One thing that might be sort of interesting is this app that the government is releasing.
Yep.
So they have this app that apparently, I don't know, 2 million people or so have downloaded already,
Yep.
which basically tells them who is in close contact to each other. So if you're within 1.5 meters of someone for 15 minutes, it's sort of recorded. And then if there's an outbreak, they will be able to track that down.
Yep.
So they do the contact tracing in like in a very good detail, which is something that China is also doing.
Yep.
So they have they have a system for telling people where they can go, when they can go there, and keeping track of each of the citizens. And I think that is probably the easiest way in this sort of suppression model to say, okay, we can let these people go, but if there is an outbreak and of local transmission, we jump on it very quickly using this sort of technology to track down all the contacts.
Yep.
And have extensive testing around that person, lock down an area, and sort of do it as this reactive process,
Yep.
as opposed to sort of letting it all go or keeping it all shut down.
Anything is getting everyone to sign up, mate.
Yeah, exactly.
People don't want to be tracked. People,
No,
and even
yeah.
though they've got all these assurances that it's on the app,
Yeah, but everyone would be on Facebook, I guess, who's probably doing the same thing anyway.
exactly. They
Yeah.
probably can track you, but
Yeah.
So yeah, it'll be interesting to see what happens with that then, because I mean, it could, that does sound pretty promising, and I think there's been a number of countries that have started doing that, hasn't there already?
Yeah. I saw at the very beginning of the outbreak, MIT released an app in the US that was doing the exact sort of thing. Same thing for contract tracing. Yeah.
Yeah. So what about like places that have done early lockdowns? Like I suppose in terms, would you say that we did go into an early lockdown? Because it really depends on when the virus gets here and how quick it's spreading, isn't it? Would you say that we went into lockdown early in terms of how far the virus had spread?
Yeah, I think so. In terms like compared to some of these other places that got out of control, definitely. And we matched South Korea very closely with our sort of curves as to when we went into when we went into lockdown. And I think because they were ahead of us, we could use them as a bit of a model to
Yep.
sort of say, okay, when are the right times to go into a full lockdown, and how much testing is required. And I think we were sort of lucky in that we are an island nation, and that all our sort of cases were imported through basically air travel. I guess through some of these cruise trips as well,
Yep.
but it was majority of these imported cases, and so closing down travel was a very effective method at sort of stopping this. So it's actually sort of unclear as to how local lockdown has stopped any sort of the outbreak. Like basically this initial curve that we saw coming out was all from imported cases. And so stopping large gatherings and closing schools and things like that, it's unclear as to if they actually had an effect on this outbreak. And schools are probably one of the things that they'll open back up first, from some recent modeling showing that kids aren't having that big of an effect on the spread of the virus.
Yeah. Yeah, interesting. So, has there, like, I suppose in other countries, is there a bit of a correlation maybe between like when they go into lockdown and sort of the mortality? Are we seeing people who don't go into lockdown, whatever you would call early,
Mm.
um, are having higher mortality rates? Is that something that they've found in the data?
Yeah, definitely. And I think that is from overwhelming hospitals, basically. So there, uh, it's not a death sentence. People can be treated for this virus, but you need to be able to treat them. And so if there are too many people to treat, there are just going to be more deaths. So if the number of infections overwhelms a healthcare system, then yeah, it's going to really start to increase the number of deaths. Yeah.
Yeah. What about like antivirals and stuff, and so treatments for it rather than actual, the vaccine itself? How's that looking? Is there stuff that's promising? Anything you've seen or that's piqued your interest at all?
Yeah, so the chloroquine stuff, the malaria drug. So it's a malaria drug that malaria developed resistance to pretty early on, I guess, but it's still used in the treatment of other sort of parasite infections and things like that. Um, it doesn't look like it's going to work. So they've done a bunch of larger trials now, and it doesn't look like this drug is actually having a reasonable effect on the outcome of patients.
Hmm.
So there was some initial talk, and Trump jumped on to it very early on and said like, "This is this miracle drug," and a couple of people died because they started taking it, or what they thought was it, and so that doesn't look as promising as was. But there is some other drugs like I think it's called remdesivir, which is a basically it's a nucleoside analog. So part of the coronavirus's RNA are these little A's, T's, or A's, U's, G's, and C's, and this one is it's like the A, but it sort of prevents the viral chain from being built out, the RNA from being built out. So it blocks the replication by interfering with the as a place of an A. So it uh, comes in, sort of blocks it, and that is looking promising. So there have been some results saying that this could work, but I think a lot of these drugs are sort of uh, last case severe treatment things. I guess
Yeah.
it's it's not like it like we have good drugs for the flu. People still die of the flu and things like that. I guess like Tamiflu is like a reasonable drug, um, but yeah, it's not like it's going to be some sort of widespread. We're going to find this miracle penicillin or something like that that's going to suddenly stop all cases. I think more likely it's just going to be something that improves outcomes, as opposed to like a silver bullet. Other
Yeah.
things like just sort of basic hospital treatment, people found that keeping people off respirators and not intubating them as long as they can, laying people in a prone position to give them more lung capacity has
Yeah.
actually been really effective in improving outcomes. So there's like sort of little things like that that people are learning, and then along with the sort of immune medication, dampening that sort of immune response at the right time, and um, yeah, and this clotting sort of problem. So getting people onto it's sort of in rare cases, but giving people the right blood thinners as well to prevent clotting in this disease. Yeah.
Yeah, I saw something recently about that with the clotting, and it's like some diseases do the opposite, um, give the opposite effect. Viruses and stuff, where it thins the blood.
Yeah.
But this one seems to be doing some like clotting and stuff. That's
Yeah. So it's sort of, I guess it's basically it's an immune mediated response. So it's from the immune system, and the body has in a balance both clotting and like anti-clotting agents, and it sort of depends which one the immune system attacks. So if it attacks the clotting agent or a factor that regulates the clotting agent, it can end up causing blood thinning, or it could end up causing more clotting based on which component of that sort of clotting cascade that it affects.
yeah. So what about like have we seen much sort of mutation with this virus? Has it changed in humans? Has there been much research out there? I did see someone send me an article about an article that came out last week saying that they found six sort of mutations or slight variations in it, and they were sort of interested to see, well, is that going to affect sort of how effective I've forgotten the word now. Like not the treatments, the what's the word?
Like the infection rate or like the transmissibility of the virus? Or
Yeah, like what? Let's just see what you've got on it. What knowledge do you have on any of these first? I'll think of the word.
yeah, okay. So yeah, this we're in the middle of a pandemic. These viruses are definitely going to mutate. That's just sort of how they work. They're this type of virus is actually sort of not very error prone. They have an error correcting system, which is unusual for an RNA virus. Generally, RNA viruses mutate much more rapidly. But as far as we can tell, there has been no sort of phenotypic effect of these mutations. These viruses aren't actually causing changes in the characteristics of the virus. These mutations aren't causing changes in the characteristics of the virus. So although we are seeing changes in the virus, and you can use that to track them, they're not necessarily increasing disease or transmissibility. Although there has been a case similar to what happened in SARS virus, where one of the open reading frames, so like a gene within the virus, has had a major deletion in it. I think this happened in Singapore, where they found this virus has been circulating for several weeks or a month or so, and that they, from studies in SARS, which also had a deletion in that same gene, that variant is probably a sort of weakened variant. So it's more host adapted. So as these viruses move from one host to another, there's no point in them killing all the things they infect because they will just die out. They need a host. So generally, viruses will adapt to the host that they infect, and they'll reach some sort of equilibrium where they can both sort of survive at some reasonable level. And so if this virus is able to outcompete the other globally circulating strain, we may end up with this sort of less virulent strain of the virus circulating. So there has been some changes within it, but nothing as obvious. I remember this initial sort of S and L type strain difference, but that was really from one paper where they were just sort of single nucleotide changes or single amino acid changes, which really didn't have any effect on the phenotype.
I've got the word. How does that affect the vaccine? Can't believe I forgot vaccine.
Yeah.
So does that change anything the way the vaccine would work, or because it's got that same?
The majority of these vaccines are targeting the S protein, which is the spike protein, so the surface protein of the coronavirus. And if the change was in that and it was significant enough, then yeah, it could definitely result in a reduced efficacy of the vaccine. But there are other coronavirus vaccines for like pigs and other animals. Maybe there's a cow one, I think. But they work. So there's no reason to expect that these would change that rapidly. So the problem with flu viruses is that they can go under this process where they have rapid evolution. They change much faster than these. And they can also shift, so they can swap large segments much more easily than these coronaviruses can. So you can get a lot of sort of mixing of these viruses, and makes it much harder to develop a sort of vaccine that will work across all the strains, whereas this one seems fairly stable, at least from what we're seeing from the genetics data. One cool website you can go to is called nextstrain.org, which is where they do this phylogenetic analysis. So they have, as all the sequences are published for these new viruses, they will incorporate them into a phylogenetic tree, and you can sort of trace them in real time as these the evolution of these viruses in real time, which was sort of the founder strains and where they've come from, and you can, and it puts them all over the globe, and they can sort of use the genetics to track back the origin. But yeah, none of that has really seemed to have an effect on the phenotype of the virus.
Yeah. So then let's go to the slight conspiracy theory then. If we can track it back, is this something, do we know definitely it's not from a lab, or like have we found that link in there that's linking it to an animal or anything specific like that?
Yeah, so the closest related virus, I think it's 96% or 92% or something like that, is a bat virus. So very closely related to a bat virus. So it's not that much of a leap to say, okay, yeah, this is a spillover from that bat virus. The same way we've seen other coronaviruses spill over from bats before. They're closely related to circulating viruses and then moved over. In the case of like SARS-1, we moved from bats into civets, maybe something else, and then into humans. And you can sort of track that that transmission period. At the moment, we don't know if there was another intermediate host. But yeah, basically, I don't think there'd be any reason to go out of your way to think that it was released on purpose, something
Yeah.
like that, right? Like there's no good evidence to say that. And in fact, it's much easier if you use like Occam's razor or something like that. The more simple solution is that yes, it behaved exactly as it's behaved 100 times before and just spilled over by animals. I mean, if you go to one of these pandemic prediction websites from 5 years ago, 10 years ago, they will all list SARS and MERS or coronaviruses as as potential pandemic pathogens. So we've known that these pathogens are on the edge of some sort of pandemic. And I guess it doesn't also make sense to release a virus that is going to go pandemic, like everyone's being infected. I mean, unless
Yeah.
it is the people in the space station that are doing it, like it's an odd sort of process to go through.
Yeah. But so is there any way that we can or we will be able to sort of definitively prove that it wasn't from a lab? Will we be able to find those like if like if there are other animals somewhere where it's gone from bats to those animals or there's something in between? Can we find that?
Yeah, I mean, if it is still circulating in those animals, and you can sort of say, like, I mean, you could always make this argument that, oh yeah, someone took it from whatever animal it's really closely related to, and then they went out and released it everywhere. Like, I mean, it depends on your definition of like made in a lab. Like, if it's just been accidentally released from a lab and sort of someone took it from a lab and they infected themselves and went for a walk in the market somewhere, I mean, I don't think we will really be able to tell that. It's the same sort of way as if it just had spilled over from someone handling wildlife or something like that, I guess.
Yeah.
But in terms of like someone genetically engineering this virus, there's no sort of signatures within the virus saying that it was touched by humans. And in fact, I've seen some stuff going around saying that the opposite is true. That there are some sequences within the virus that are sort of unknown to people, and so it would be very unusual for people to be able to know what those sequences were and then put them into the virus.
Yeah, cool. There we go. No conspiracy. I'll believe you on that one. I'm going to ask some fun questions. We do this at the end all the time. And if we think of anything else while we're there, maybe if there's anything you want to maybe talk about or let people know, we can do that at the very end. So first question I give everyone. I'm a single man at the moment in this pandemic.
Yeah.
First date ideas, mate. Social distancing. First date ideas. Have you got anything for us?
Currently,
Yeah. Any ideas?
oh geez, man,
I'm
it's
putting
sort
you
of
on the
hard. Like
spot.
the Zoom date. One of my mates went and met up with someone on the Strand, but had it's sort of the walking at a distance situation's probably not much better than a Zoom date. But yeah,
Yeah.
it's hard to do at the moment.
It's hard
The
being single.
general all the sort of local spots of bars and cafes are not there.
Yeah. Yeah, you can't really do anything social, so it's like, yeah, like you
Yeah,
said, Zoom or go for a walk.
yeah, exactly.
Got to get creative.
Yeah.
So next question, because we've obviously had some toilet paper issues around the country and stuff. Not really anymore, I don't think. But are you a scruncher or a folder?
I am a folder. Yeah,
Folder, folder
yeah.
for life.
Yeah, I was very lucky in that my mom sent me a box of toilet paper right in the middle of the of the outbreak.
Yeah.
She got the Who Gives a Crap subscription,
Oh,
so
yep.
individually wrapped rolls turn up just when I was like one roll away from being in trouble.
Yeah.
Uh, yeah, folder.
That's good. So I suppose that leads on to the next question: is like, what do you have lots of in supply? Whether it's something you went and panic bought on purpose or you just happened to get it because there was nothing left at the shops. Is there anything you have in large supply at your place?
Oh yeah, I got a bunch of Easter eggs, I guess, which I like that, and rapidly bought the day before Easter. Um, what else do I have? I didn't try. I was trying to like very early on. I was like, oh, don't panic buy anything. I went and
Yeah.
bought a couple of extra beans of things. I get the Dinnerly delivery dinner box thing sent.
Yeah.
So that's a good way to sort of avoid the shops, and it hasn't had any sort of supply chain issues.
Yeah.
I went and bought a bunch of um, filament for my 3D printer. So I've been sitting at home 3D printing stuff,
What
which
have you
is
been
still.
printing?
I've been making little figurines, and I made a cast to make pot plants. So you
Oh,
like
cool.
basically print out a cast in like it's like a little shape of a house, can fill that with some like plaster of Paris or concrete or something like that, and
Ah, yeah.
produce like little pots and things out of it. Just sort of boring things that you do in
Yeah.
isolation.
No, that's cool, man. That sounds fun. And then, so what have you got that you don't have much of in supply that you're like, oh, I'm going to run out of that, or I don't have much of that at the house?
Oh, coffee. I just
Coffee.
saw this too before. Yeah, I've got about one scoop left. Yeah,
Oh,
make
that's
it that.
rough.
Yeah,
Especially if you got to get that thesis done, mate.
yeah, exactly. Yeah, I've been
Yeah,
trying.
smashing the coffee. And so after this is all over, what's the first thing you want to do when we're when we're allowed out to play?
Oh, I feel like it's going to be a really slow release, and I'm
Yeah,
like, yeah, like if it was all done tomorrow, I guess maybe I'd go to some mosh pit somewhere or something like that. But
yeah,
yeah, I feel like it's going to be a very slow sort of. I'll creep out to a bar one day and be like, "Is this okay?" Um, but yeah, maybe hopefully I'll just wake up or realize one day that I'm sitting in a bar and not worried about the person who's coughing near me or something like that.
yeah, yeah.
Yeah.
And so another like last sort of fun question, I suppose it's you know, a lot of people out there, not much to do. Any recommendations of a good book, good game, movie, or TV series that you enjoy? Something that people can maybe do to pass the time and sort of,
Oh yeah. So I've been reading The 100-Year-Old Man, which is a good book.
yeah,
I got a, a Super Nintendo emulator for my computer, so I've been playing Pokemon Fire Red and some Donkey Kong, which is good.
yeah, sweet.
And yeah, I think I've been watching Kidding on Stan, which is like the new Jim Carrey series. It's kind of dark,
Yep,
but yeah, it's good in a way. I mean, you don't want to watch too many episodes in a row, I guess. But
yeah.
yeah, it's good.
Yeah, cool. Um, and so I suppose that's all the fun questions we've got there. But I just want to know, is there anything you've seen that you've loved that's innovative or really interesting that you've found? And this can be doesn't have to be in the academic world, although it could be. I suppose we sort of talked about that with the being able to print the vaccine.
Right.
Um, is there anything you've seen that you're like, oh, that's awesome, that's really really clever, or anything around?
Yeah, like I liked all the sort of if you go onto like Thingiverse, so I do a lot of 3D printing stuff, and so if you go on there, there is just a bunch of people uploading designs for different medical equipment, making their own masks, developing. There was a big story about people developing components for respirators, or and then printing them out and giving them on. So yeah, there's sort of a lot of this sort of maker community is sort of giving back a lot and sort of saying like we have this production within our households, and they're producing like a lot of material that can be used by people who need it. Yeah, pretty cool.
Yeah. Yeah, I've got a mate who's actually he's making the the band part that goes on the visors
Yeah,
for the face shields. So yeah, he's I think he's like, "Oh, I can only print like 10 a day, but that's 10 more than they had." So
yeah, exactly. No, it's
yeah.
great. And then you think there's another 100 of him doing it, like it's a
Yeah.
whole world.
Yeah, that's awesome. And I've seen other places like tooling up for different things. Companies who have big vats and stuff to make hand sanitizer and stuff like that. Like that's been really interesting to see,
Yeah,
hasn't it?
I saw just in the news today that Stephen Hawking's family donated his respirator to the NHS. It's just like it's so
Yeah,
sweet. Yeah,
that's random.
yeah.
Yeah. Yeah, cool. Well, is there anything else you can think of that's interesting or to do, with the virus? I'm sure I'll get off this call and instantly think
Yeah.
of like 10 things I want to know, and I'll message you. But is there anything else that you think it's important for maybe people to know, or that you want to
Um,
tell people about this virus?
yeah, I guess I mean this has kind of come out a little bit now, but that there's no predictive power in trend lines. So initially, everyone was plotting these exponential curves with a trend line in it, saying, in 2 months there's going to be 100 million people dead or something like that, and sort of just plotted that out. And I think it's in sort of important part of like epidemiology and modeling that you can't actually the date doesn't predict number of cases. So people sort of make these sort of trend line analysis, and it created a lot of fear. And I think that actually drove part of this panic buying scenario that we saw. Whereas like using actual epidemiological models gives you a much better idea of how the disease outbreak is going. So I think, yeah, trust experts and yeah, stay inside, don't panic.
Yeah. Yeah, cool. Awesome, man. Well, thanks for the chat. It was really good to get your take on some of that and some of the info. Just before we go, what's your thesis on? Give us the specifics.
Oh yeah, so my thesis is on the pathology and epidemiology of rhinovirus infection in Australian freshwater turtles. So basically, we went around conducting a survey of this viral disease in turtles throughout northern Queensland, and then sort of developed an understanding of the pathogenesis of this disease. So different factors that influence how the disease develops in a way of sort of so our turtles are very different to other turtles in the world, and so they're not we're not sure if they are actually dying from the disease because people aren't looking, and whether or not they uh, whether or not they produce the same sort of pathology, or that we don't really have the test to do it. So it's sort of this broad topic on pathology and epidemiology. Yeah.
Yep. Yeah, cool. I'm going to quickly just go to Facebook and see if we've got any questions on there. I haven't done this before, with the live, but let's see what we got. Let's see if we got anyone in there. Almost need an assistant to
Yeah.
be here and do it for me. I need a young Jamie. That'd be great. There we go. Grow a mo. Everyone's just bagging me about my mo. Got nothing on yours, mate. All right, well, we might leave it there. Thanks, man, for coming on. Really great chat, and thanks for all the help with fact checking all the articles that I've
Yeah,
been
no
sending you.
worries, man. I'm glad we
Um,
could finally figure it out. Yeah, sweet.
yeah, that's awesome. So yeah, thanks, man. And who knows, we might get back on again one time and have another chat down the track once we know a little bit more, and you can
Yeah,
maybe give us some more info then.
definitely. Hopefully, looking back from a bar somewhere when we're all
Yeah,
free.
we'll do it out somewhere at a bar. That sounds
Yeah,
good. We'll have a few beers or something. That's
exactly.
good.
All right.
Awesome. All right,
Yeah.
mate. Have a good one. Thanks for coming on the show, and I'll talk to you soon.
Cheers.
There we go, guys. Great episode. Thanks to Wytamma for coming on the show. We've talked about afterwards. I got to go up to Townsville and have a beer with him. So we're going to make sure when this thing's all over, definitely going to head up there and have a beer with him. So like we said as well, we might sit down and have another chat at some stage. Get him back on the show, maybe when there's some more research, or maybe when when the vaccine is closer to being sort of being used. We can talk to him about how that's going. So thanks to him for coming on the show. Make sure you guys like and subscribe. Share this episode around. Subscribe so you can stay up to date with all the episodes as they're released. Hopefully have another Beer With episode as well coming soon. So stay tuned for that one as well. Make sure you're following me on all platforms at BoyKingKoi. You can go to the website, abeerwith.com. There's links to everything there. Come join us for Friday knockoffs, 6 o'clock Friday. That's Australian time, where we just basically get together on a web chat, have some drinks, and kick back and relax. So I'll see you guys there maybe on Friday. All right, guys, that's it for this episode. I hope you're having a great day or night, wherever you are, and I'll see you guys soon for the next episode.